everyone, my name is Bailey Bashara. I'm a biology major and I completed my capstone presentation on the effects of cortisol on blood coagulation in zebrafish. Blood coagulation is regulated via the coagulation cascade, a series of reactions in which an inactive factor becomes enzymatically active following contact with the previous enzymes in their activation pathway. Clotting factors are the enzymes which activate each step of the clotting cascade, with 12 factors named factors 1 through 11 and 13 in total. The clotting cascade can be triggered via two pathways: the tissue factor pathway, also known as the extrinsic pathway, in which coagulation factors 7 and 10 are activated by platelet tissue factor, which is released by blood vessels under certain pathological conditions. It can also be activated via the contact pathway, also known as the intrinsic pathway, in which factors 12, 11, 9, and 8 are activated by vascular trauma. As you can see in Figure 1, the intrinsic and extrinsic pathways converge to form the common pathway after the activation of factor 10 by either factor 7 in the extrinsic pathway or factor 9 in the intrinsic pathway. Following this convergence, the activation of factor 13 results in the formation of fibrin proteins, which bind together to form a blood clot. There are mechanisms which inhibit the coagulation cascade once the amount of needed coagulation has been reached. The inability to inhibit coagulation when needed, or premature initiation of the coagulation cascade, can lead to blood clotting disorders and dangerous clot formations such as pulmonary emboli. This means that the precise regulation of the coagulation cascade is extremely important. When the coagulation cascade is unregulated, coagulation disorders occur. Coagulation disorders due to deficiencies in coagulation factors can result in excessive bleeding, often causing joint and muscle bleeds. These disorders can be inherited, such as hemophilia, or they can be acquired later in life, such as thrombocytopenia. Other dysfunctions in the coagulation cascade can cause hypercoagulable states. The susceptibility to these states can also be inherited or acquired. A hypercoagulable state can lead to dangerous clot formation such as deep vein thrombosis and pulmonary emboli. In such a case, a thrombus builds in a deep vein due to the inability to inhibit the coagulation cascade, known as deep vein thrombosis, and this large thrombus has the ability to break off and travel to the lungs, known as pulmonary emboli, which can be fatal. Both types of coagulation disorders can be fatal, requiring treatment to re-regulate the coagulation cascade. Zebrafish carry the same coagulation genes and factors as mammals, providing evidence that the coagulation process is fundamentally the same as in mammals. Zebrafish, or Danio are a telos species used as a genetic model. Pictured in Figure 2 is a male zebrafish, which is pictured on the top and is slender and more golden in color. On the bottom is a female zebrafish, which, as you can see, is a bit larger. Like in mammals, all coagulation factors are found in both blood and liver cells, and the coagulation cascade in zebrafish is activated by both intrinsic and extrinsic pathways. This high similarity indicates zebrafish are a suitable model organism for research on hemostasis and thrombosis generation. In humans, several hormones have been shown to increase the likelihood of coagulation and the risk of thrombosis. The natural estrogen present in females, estradiol, has been shown to increase the likelihood of hypercoagulable states. Sex steroids such as progesterone and 17-alpha-ethinestradiol, which are commonly found in contraceptive pills, are known to increase likelihood of thrombin formation in women. In zebrafish, embryonic exposure to excess sex steroids has shown significant increase in the transcription of genes encoding for coagulation factors and can lead to activation of the extrinsic pathway of coagulation, indicating that zebrafish react to hormones similarly to humans. Cortisol is the glucocorticoid hormone which mediates the stress response in many vertebrates, including humans and zebrafish, and can promote clotting, indicating that stress can induce the coagulation cascade in those with and without existing coagulation disorders. A study on post-traumatic stress disorder found that patients with PTSD had higher levels of clotting factor 8 and fibrinogen, which can elicit hypercoagulability. Additionally, research has shown that those with Cushing's syndrome, which causes an excess level of cortisol, have higher incidence of thromboembolic complications. Studies have also shown that the synthetic glucocorticoid clobetasol, a stress hormone, upregulates genes F2, F7, and F9b, which code for coagulation factors 2, 7, and 9 in zebrafish. Ultimately, if increased cortisol levels in zebrafish cause upregulation of the genes coding for coagulation factors and therefore a hypercoagulable state, zebrafish could be used as a model organism to test anticoagulant medications. This would be beneficial in the discovery of treatment of thromboembolic events associated with stress-related diseases such as Cushing's disease. I hypothesize that, as in humans, an increase in cortisol due to stressful environment induces transcriptional upregulation of genes encoding coagulation factors in zebrafish, predicting higher transcripts of F7 and F10 in stressed fish and higher transcripts in stressed females than in stressed males. The objective of this study was to investigate the effects of cortisol on blood coagulation in zebrafish via the effects on transcription of the genes coding for coagulation factors in chronically stressed fish. As you can see in Table 1, we measured the transcript levels of gene F7, encoding coagulation factor 7, and initiates the contact pathway of the coagulation cascade. We also measured the transcript levels of gene F10, encoding coagulation factor 10, which initiates the conversion of prothrombin into thrombin, leading to the formation of a blood clot. In this study, we used 18 male and 18 female zebrafish in tanks of 6 by sex and subjected them to acute stressors repeatedly for 7 days. These acute stressors included the crowding of 6 fish in a 250 milliliter beaker with 150 milliliters of water for 50 minutes, 3 consecutive net captures, chasing the fish with a net for 8 minutes, and reducing water levels to expose the dorsal body wall for 2 minutes. The times at which these stressors were administered were changed daily to prevent the fish from becoming habituated to the stress events. The schedule of stress events can be seen here in Table 2. Additionally, 18 male and 18 female zebrafish were used as control and not exposed to the chronic stress environment. 24 hours after the completion of the last stress event, all fish were euthanized and dissected. The livers were immediately transferred to trireagent and stored at -80°C to be used for RNA extraction. In RNA extraction, RNA was isolated from each liver using the Zymo Research MicroPrep Direct-Cell RNA kit, and cDNA was synthesized from the isolated RNA using the Maxima H- cDNA synthesis kit and stored at -80°C to be used for qPCR. Transcript levels of the coagulation factor genes F7 and F10, as well as the housekeeping gene beta-actin, were measured in triplicate for all cDNA samples using CyberGreen Master Mix. For data analysis, transcript levels of F7 and F10 were normalized to beta-actin and calculated using the double delta CT method. A t-test was then used to test for differences in relative transcript levels between the stressed and control groups and between the sexes using an alpha of 0.05. Figure 3 on the left shows the transcript levels by experimental group. Mean plus or minus standard error of the normalized delta CT values can be seen for both the control and stress groups, on the left for factor 7 and on the right for factor 10. Stressed and control groups for each gene had sample sizes of 34. For factor 7, mean transcript levels of the control group was -0.211, and for stressed groups, -0.367. And in factor 10, -0.1 for control groups and -0.576 for stressed groups. A t-test showed no statistically significant difference in transcript levels for either F7 or F10 between control and stressed groups. Figure 4 on the right shows the transcript levels by sex. Control and stressed females for each gene had a sample size of 16, and control and stressed males had a sample size of 18. For factor 7 on the left, control females had mean transcript levels of 0.063, and stressed females -0.583. Control males -0.063, and stress males -0.004. Transcript levels of factor 10 control females was 0.051, stress females -1.361, control males 0.1, and stressed males 0.122. T-test showed no statistically significant difference in the transcript levels of either gene between the sexes independently or between stressed females and stressed males. In conclusion, transcript levels did not differ significantly for either gene between stressed and control groups or between the sexes. There is no evidence that increased cortisol causes differences in the transcription of genes coding for coagulation factor 7 or 10. However, high variation among samples resulted in low statistical power when performing the data analysis. Further experimentation could be performed using a longer period of stress events or investigating the transcription of different coagulation factors in the coagulation cascade. I would like to thank the College of Arts and Sciences as well as the Honors Program for providing the funding for this project. I would also like to thank my faculty sponsor, Dr. Baker, for her guidance. Thank you for watching.